Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic genetic code and RNA types. So this is from the chapter molecular basis of inheritance. Here you'll learn the entire concept of RNA and genetic code. So without any delay, let's start with the topic. So let's start with the topic genetic code. So what is a genetic code? It is a sequence of nucleotide. Okay, so it is a sequence of nucleotide that is a nitrogen basis in mRNA that contains information for protein synthesis, that is translation. The sequence of three bases determining a single amino acid is called codon. So what is a codon? It is a sequence of three bases. Okay, so three bases combine to form a codon. So you can see the codons below. You can see one mRNA strand is from 5' to 3' region, and you can see the codons three in pairs. Okay, you can see 3 1 2 3. So here total 9 codons are here. So three bases or three nitrogen bases combine to form a codon. Now George suggested that for coding 20 amino acids, the code should be made up to 3 nucleotides. Thus, there are 64 codons. So if one codon is formed by 3 nitrogen bases, then there are total 64 codons. Hargobind Khurana developed the chemical method in synthesizing RNA molecules with defined combination of bases, that is homopolymer and copolymer. Marshalls developed cell-free system for protein synthesis. Severo osea enzyme, that is a polynucleotide phosphorylase, is used to polymerize RNA with defined sequences in a template independent manner. So all the three scientists have said different things, so you can see there. Next you can see the genetic code. There are 20 types of amino acid involved in translation. One, you can see the names: alanine, arginine, asparagine, aspartic, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine, and all the short forms of the abbreviation symbols. Okay, not abbreviation, the symbols are given in the bracket, like alanine, its symbol is ALA. Okay, short form. Arginine, uh, short form is ARG. Okay, so this 20 amino acid you have to memorize. Now you can see the code dictionary. You can see here in the right hand side you can see the columns UCAG, left hand side also UCAG, again this side UCAG. So this is the entire genetic code. Okay, so you check first one. You see UUU, that is codes for phenylalanine. Okay, UUC, that is phenylalanine. Next UUA, that is for leucine. So here are 20 different amino acid. It forms a genetic code. Now these are the silent features of genetic code, like codon is triplet, that is 3-letter code. There are total 61. Okay, so 61 codon code for amino acid. So 3 codons, that is UAA, next is UAG and UGA. So there are 3 codons you can see. So out of the 64 codons, these are the 3 codons, that is UAA, UAG and UGA. Okay, so these 3 codon acts as a stop codon. So these acts as a stop codon, that is termination codon or nonsense codon. So total 64 codons are there. Out of that 3 are stop codon, that is UAA, UAG and UGA. And AUG, okay, AUG has a dual function. It codes for methionine and acts as an initiation codon. So UAG is a start codon, as well as it is called an initiator codon. So in eukaryotes, methionine is the first amino acid, and formylin methionine in prokaryotes. Okay, so in eukaryotes, methionine is the first amino acid, and formylin methionine in prokaryotes. So an amino acid is coded by many codons except AUG for methionine and UGG for tryptophan. So such codons are called degenerate codons. So you can see when strands is given, that is from 5 prime to 3 prime, you can see the initiation codon or initiator codon AUG. All are triplets, you can see, and stop codons you can see UAA clearly given. So next we see again we see the silent features of genetic code, like first one is no punctuations between adjacent codons or commaless code. The codons is read in mRNA in a contagious fashion. Okay, so genetic code is universal. For example, for bacteria to human, UUU codes for phenylalanine. Some exceptions are found in mitochondrial codon and some protozoans. So genetic code is non-overlapping, and genetic code is unambiguous and specific. That is, one codon specifies only one amino acid. Okay, so you can see in the codons there are 3 nitrogen bases. They are commaless there. That means no comma is there. Okay. So next we see mutation and genetic code. So relationship between genes and DNA are the best, okay, are best understood by mutation studies. So deletion and rearrangement in a DNA may cause loss or gain of a gene and so a function. So insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. It is called frame shift insertion or deletion mutation. Okay, so you can see the normal sequence you can see is in the middle. Okay, and insertion of one base is given on top. You can see G is added. Okay, and below one, last one, you can see deletion of one base. Okay, so you can see the entire reading frame is changed. So on top you can see insertion of G, middle one is the normal strand, and on top one nitrogen base is added, and below one nitrogen base is deleted. Okay, so you can see on top G is added, insertion of one base, that is G. So entire sequence reading frame is changed, and below you can see one is deleted, that is A is deleted. So entire reading frame is changed. Okay, so insertion, that is insertion or deletion of 3 or multiple bases. Insert or delete one or more multiple codon. Okay, one or multiple codon. The reading frame remains unaltered from that point onwards. Hence, one or multiple amino acids are inserted or deleted. So it proves that codon is triplet and read contagiously. So you can see here middle one is the normal sequence, and you can see on top insertion of 3 bases, that is GAG. So again I repeat, you see in this middle, that is a normal sequence, where in the top strand you can see from 5 prime to 3 prime, insertion of 3 bases are added, that is GAG. Okay, you can see the reading frame is altered. Okay, if so you can see GAG is added. Okay, and next below you can see 3 bases are deleted. Okay, that is ACG is deleted. So you can see the reading frame. All right, so again I repeat, middle one is the normal sequence, on top 3 nitrogen bases added, the GAG, and in below 3 nitrogen bases deleted, that is ECG. Now moving to RNA world. So we know that there are different types of RNA. You can see messenger RNA, that is mRNA, ribosomal RNA, that is rRNA, and transfer RNA, that is tRNA. So RNA was the first genetic material. It acts as genetic material and catalyst. Essential life processes like metabolism, translation, splicing, etc. involved around RNA. So DNA involved from RNA for stability. Now you can see the different types of RNA are there. Messenger RNA, that is mRNA. So it provides template for translation, that is protein synthesis. Next is ribosomal RNA or rRNA. The structural, so it is the structural and catalytic, it plays a structural and catalytic role during translation. That is example, you can see 23S rRNA in bacteria acts as ribozyme. Okay, so 23S rRNA in bacteria acts as ribozyme. Next is tRNA, that is transfer RNA. Okay, so it brings amino acid for protein synthesis and reads the genetic code. So you can see the mRNA diagram, the ribosomal RNA and the transfer RNA, all the diagrams are given. Next, you can see in the types of RNA, the tRNA I have explained. You can see Francis Crick postulated presence of an adapter molecule that can read the code and to link with amino acid. So tRNA is called adapter molecule because it has an anticodon loop that has bases complementary to a codon, then amino acid acceptor end to which amino acid binds. Next we have ribosome binding loop and enzyme binding loop. So you can see all the loops are given. On top you can see in the picture right hand side amino acid acceptor end, then ribosome binding loop, then anticodon loop and enzyme binding loop. So you can see for initiation there is another tRNA called initiator tRNA. There are no tRNA for stop codons, so stop codons don't have a tRNA. So secondary, that is 2D structure of tRNA looks like global leaf model. So you can see right inside is a 2D structure of tRNA, that is a shape is global leaf model, and 3D structure looks like inverted L shaped. Okay, the 3D structure looks like inverted L shaped. You can see the pictures given. So you can see the pictures how tRNA is helping in process of translation of bringing the amino acids. Okay, so you can see one tRNA bringing tyrosine and another serine. So 3D structure looks like an inverted L shaped. Okay, so 3D structure of tRNA looks like inverted L shaped. So thank you student, this was all of the entire video lecture. You have discussed about the genetic code and different types of RNA. So stay at home, stay safe. Thank you students.